Good morning, everybody. Thank you for coming to our first lecture of the spring 2016. Dr. Coy Heldermon is a professor at the University of Florida. He's also a doctor, a physician. He has an MD, Ph.D., and he graduated from the University of Oklahoma. And I don't know where you did your undergrad, but he'll talk more about his scientific career during his talk. He did his medical school also at the University of Oklahoma and his residency at the Washington University in St. Louis. His specialty is internal medicine and oncology, and that is cancer medicine. And we are very happy that you're here, and welcome.
Thank you. Thank you. So, the title of my talk is Many Roads, One Goal. I hope that I convey that there are a lot of different ways to actually get to where I'm at, but more importantly, to get where you want to go. So where I come from, I grew up in rural Oklahoma. It was 19 miles from my house to the nearest town on a cattle ranch. This was my hometown, Chelsea, Oklahoma, and that is it. You can see all of it. Okay, that was the nearest town that was 19 miles away, population roughly 2,000 people, and the median household income in Chelsea is about 24,000. So to give you, that's the median household income now, not many years ago. It's about 70% Caucasian, 25% Native American population. My senior class had 62 students. My dad worked part-time on the ranch, but most of his income came from oil and gas delivery, and then later as a salesman. He had a bachelor's degree. My mom finished her bachelor's degree when I was in late junior high, and she worked as a clerical secretary for essentially all of my upbringing until I went to high school. And this is the house that I lived in for high school. Prior to that, we were in a trailer house, but the barns were there even then. So I did my bachelor's in animal science at Oklahoma State University in Stillwater. I did do work study when I was in school. Initially I worked in a lab where the entirety of my job was to take cow blood and isolate the DNA from it. And that was for a guy who was doing research on twinning, trying to find loci that were associated with twinning in cows to see what causes that. And then he lost his funding, so I went to a different lab that was in the food science department, and they were working on figuring out better ways to make milk digestible to people who can't digest milk. There's this stuff called sweet acidophilus milk, and the founder, or the head of the food science department, that was his baby. He discovered it, and so we were trying to make a superbug who would make even more of this enzyme beta-galactosidase that breaks down the sugars in milk, which is what people who are lactose intolerant can't break down. So that was my project for much of my undergrad. And then I went to OU, which, imagine if you went all of your undergrad at FSU, and then you went to UF. Okay, it's kind of that same kind of gestalt, and did the MD PhD program. So the MD PhD is you do 2 years of the medical degree, then a PhD, and then you finish the next 2 years of the medical degree. And my PhD thesis, or dissertation, was topologic and mutagenic studies of the hyaluronan synthase from Strep pyogenes. How's that for a mouthful? So topological just means this is a protein that sits on the membrane of a cell, and it makes this stuff called hyaluronan, which is a nice thick sticky coating that this bug puts around it so that our immune system can't recognize it, protects it from being degraded. And this is the flesh-eating disease bug, or one of the flesh-eating disease bugs. It can also cause strep throat. and this protects it from your immune system so that it can keep you sicker longer, essentially. So I was studying how that protein fits in the membrane and then mutating it to see if I could change how long the chains and what parts of the protein are important for production of hyaluronan. During my whole impetus for doing the MD Ph.D. was when I was in when I was in junior high. I think it was my grandma had given me a subscription to National Geographic, and they had this article on cloning. And up until then, I had aspired to be the world's first honest politician. I think about that time I actually figured out that that wasn't really possible to be a successful, honest politician. And so this article really captured my imagination. And it was at that point that I knew that my life's goal would be to develop gene therapies for disease. And so that was kind of the track I set myself on as my goal. Most people who want to do that as a goal don't go to animal science. They go to biology. But animal science is where I got a scholarship. Animal science is where my dad had his bachelor's, and so I was familiar with many of the professors because they'd taught my dad way back in the day. But to do gene therapy of disease, you got a lot of ways and a lot of points that you can do that. And so coming out of my MD PhD, I was a little undecided. So I interviewed in pathology, obstetrics, and in internal medicine, because they were all three different ways that you could get to doing gene therapy. You can also do pediatrics or neurosurgery or whatever. But obstetrics is where the majority of genetic diagnoses actually happen in the US. Pathology is very conducive to doing research because you have samples from everybody. And internal medicine is kind of of the medical professions, is the sit down and think about it field, so to speak. Surgeons cut, and obstetricians catch, and internists, they sit and ponder. Okay? So I interviewed in all of these, and we have this thing called the match, where you interview and then you rank the programs that you interviewed in the order in which you preferred them. And the programs do that for all the people that they interview, and then the computer meshes those together. And I had ranked my top choice as internal medicine at Washington University in St. Louis and matched to Stanford OB. So I did an intern year at Stanford in obstetrics. And I thought, if there's any place on the planet where you can do obstetrics and do research, it's Stanford. And I learned that that's true if you're a reproductive endocrinologist, but if you're wanting to do gene therapy research, it's probably still not wholly supported in an obstetrical field, because those darn babies don't deliver at regular hours, right? You can't say, I'm sorry, I'm in the middle of an experiment, you're going to have to wait. Don't push, right? It doesn't work very well. And so I told the program director that I think that I need to go to a different route so that I can do the research that I wanted to do. And so they called the internal medicine residency program at WashU, which was my original #1 choice, and they said, sure, come on over. So that's where I went. And I was part of the physician scientist training program, which is basically for people who have MD PhDs. It's a track where instead of doing a 3-year residency and a 3-year fellowship, you do a 2-year residency and a 4-year fellowship. Not saving you any time, but during fellowship they protect time for actually doing research at the same time. And so I did my internal medicine residency and oncology fellowship at WashU. And then I stayed on for a year as an instructor and then came here. And I learned, because when I started this oncology fellowship, I really thought I was going to be a hematologist because sickle cell disease, cystic fibrosis, all these monogenic, meaning one gene causing a diseases, hemophilia, thalassemia, all of these are diseases that hematologists primarily treat and are probably most amenable to gene therapy kind of interactions. But there is the aspiration and then there's the reality. And the reality was when I cared for breast cancer patients, I really enjoyed doing that. And less so when I was taking care of the hemophiliac. And so I had a concentration in breast. And while I was at WashU, I found a gene therapy lab because that's what I have always wanted to do. And that lab works on lysosomal storage diseases. There were 2 gene therapy labs at WashU, and they both worked on lysosomal storage diseases. And working on those diseases and the approaches to those diseases, again, was my passion. And so when I came here because UF is one of the top 5 centers in the US for AAV-based gene therapy. AAV is a type of virus that's been altered so that we can use it to deliver genes to correct things. And the people who discovered AAV, modified AAV, figure out the structure of AAV, have actually used it in human clinical trials, are here. So it seemed like a pretty good place to be. And they've allowed me to have both the breast clinic as well as the lysosomal storage disease clinic. Personally, I met my significant other during my last year of residency. I would say that the heart wants what the heart wants, but putting off personal life a little bit has its advantages professionally. We have a total of 4 kids, ages 5, 7, 14, and 17. She works full-time, getting her master's degree at night. I grew up in a Christian tradition, and I'm a Christian, and I volunteer at Mobile Access Clinic. So between this and the 4 kids, that's pretty much everything outside of science and medicine, okay? Get used to it. I know you all have these wonderful social lives outside. That all changes once kids come into the picture, okay? And it's a good trade-off, but just to give you warning. So I'm going to briefly go over the kinds of research that we do in my lab. One aspect is this hoping to work with a natural phenomenon, which is maternal-fetal microchimerism. When you were in your mother's womb, she gave you some of her stem cells, and you gave her some of yours. It happens. That maternal-fetal barrier at the placenta isn't absolute. The interesting thing about that is, especially if you're the last child, I can assay you in your 20s to 30s to 60s, and if I know the specific markers that are specific to your mom that she didn't give you genetically, I can still find some of her cells in you, and vice versa. Okay? Unless, of course, you were the first child. Later children, for some reason or other, their stem cells wipe out the prior child's stem cells. Okay, so you won that battle of sibling rivalry. And what I want to do, or my concept, is that for these rare inherited diseases, a lot of time what's going on is you're deficient in a protein or an enzyme, and you don't need that much of it to be normal. Most of our biologic processes, we make more than enough for what we need. We make enough for backup. And for hemophilia, for example, if you have 2% activity, it's the difference between being hospitalized 6 times a year and not being hospitalized at all. The difference between requiring weekly or 3 times a week injections of enzyme and not requiring any enzyme replacement except for every few years. Just 2%. It's a big deal. So my hope is that we can develop ways to enhance this sharing of maternal cells with fetal offspring so that mom cells will provide enough of the enzyme that the child won't be severely affected. Does that make sense? And so I use a lysosomal storage disease model, a disease called MPS 7. And it's handy because the enzyme that is deficient is very hardy. You can put it in formaldehyde for 6 hours and it still works. You can leave it out at room temperature on a bench for a week or two and it still works. And most other things in our body sort of turn to mush and stop working at that point. And we have this wonderful substrate that it converts into red, so you can find one cell in a body. Because you add the substrate, turns that cell red. Okay? So I've been working on ways to use this system to determine what cells are getting from mom into baby. Okay? And so what I've seen is that you can see that there's muscle cells and fat cells and liver cells and lung cells and skin cells and bone cells that all came from mom as rare events. And we've been using these things called cytokines, which are signals that our body makes to communicate between cells to tell them to do things. And there are certain kinds of cytokines or growth factors that cause higher production of stem cells or cause stem cells to move from bone marrow to blood, which hopefully is where they can get across the placenta into the offspring.
I have a question.
Mm-hmm.
So the mothers are going to have these babies, but the baby is carrying a disease, so they have been screened, but the mother is normal herself?
Correct.
Okay.
Correct. So by definition, if she's a mom, she doesn't have one of this disease. There are several of these lysosomal storage diseases where you don't live long enough to have children. And so what I've been able to show is that with certain cytokines, you can go to finding almost no red cells to finding that 20% of the slices have at least a cell, which sounds like a big jump, but it's one cell out of a slice. Okay, so it's still, we're probably going from 1 in 10 million to 1 million to 1 in 100,000, which isn't quite to that 1 or 2% range. But with some of our cytokines, we're seeing a dose response, and so we're doing combinations of cytokines to see if we can continue to get up to 0.11%. That's our goal as a proof of principle to move forward to try and use this practically. Does that make sense? Kind of? So the primary disease that I work on, though, is Sanfilippo syndrome, type B. There's A, B, C, and D, and now there's even an E, Sanfilippo syndrome. And they each are a deficiency of a different enzyme. And this is a very unfortunate family. So this is autosomal recessive, which means that you need 2 copies of the bad gene in order for it to affect you. So there should be a 1 in 4 chance that an offspring of 2 people who carry 1 bad copy of the gene will have an offspring. This unfortunate family had 4 children that are all affected. So generally, somewhere between the ages of 2 and 5, they have been developing seemingly normally, and they start not keeping up with their peers reaching developmental milestones, So these are kids who are all running around, talking, playing, interactive, and then they start not growing quite as fast, not picking up, sentence lengths, math concepts as well. And then they start to regress. So mom usually notices something is wrong around 2, 3, or 4, goes to her pediatrician, and between the pediatrician initially thinking mom's being paranoid, to the pediatrician saying, yeah, we're definitely falling behind, to finally getting a diagnosis is usually about 4 years. Okay? And somewhere between, from around 5 to around 10, they progress from being somewhat normal down to essentially being wheelchair-bound, non-communicative, being complete care. And they also, so similar to these individuals. And then somewhere over the next 10 years, they generally die of heart failure, pneumonia, et cetera, because they can't protect their airway when they eat. So they do what we call aspirate, which means they essentially swallow, instead of swallowing, they breathe liquid, food, etc. They get an infection and that kills them. Generally, the average age is somewhere between 15 and 20. Okay. So I use mouse models of most of my diseases as kind of the proof of principle. This is the average survival of the MPS IIIB mouse that's not treated. And with, if I give, and this is a wild type mouse, and if I give either intravenous gene therapy, or it looks like this. If I give intracranial gene therapy, it looks like this. And this is the number that are surviving at different time points. So 50% survival of about 300 days versus 350 versus 420 versus 600 if I give both intravenous and intracranial. We're still not quite up to normal. So we're working on different ways, finding which viruses do better delivery, which injection sites do better, delivery and correction of disease. So that's the lysosomal storage disease side. On the other side, I do breast cancer. So about 5% of my clinical population is lysosomal storage diseases because they're rare diseases. 1 in 60 to 100,000 kids who are born have these diseases. There's breast cancer, about 1 in 8 women will develop breast cancer in their life. So number of patients is a bit disproportionate. And consequently, about 95% of my clinical population is breast cancer. One of the things that you may or may not be cognizant of is the big debate that's going on about screening for breast cancer. So all this changing guidelines on mammography, when to start, how often to get them, whether we should do it at all, right? And part of the problem is mammography machines aren't cheap, and you need a radiologist to read them. And they're not terribly sensitive or specific for breast cancer. If you take 1,000 women and start them getting annual screenings at age 40, which is the American Society of Clinical Oncology recommendations, then by the time she's 50, out of those 1,000 women, 500 to 600 will have been called back because there's something suspicious. A little bit of anxiety there. Okay, out of that 600, roughly 200 are going to get an ultrasound and 100 are going to get a biopsy to find the 10 to 20 that actually have breast cancer. Okay, so that's a lot of anxiety, a lot of procedures, a lot of steps along the way. So we need something that will help reduce the number of second calls, the number of false biopsies or biopsies that aren't breast cancer. And so what I'm doing is I'm taking some breast cancers from women, putting them in mice and growing them, which works about 10% of the time, maybe, depending on the tumor type. And then we're comparing the blood composition of those mice to mice who aren't growing breast cancers, and look for human proteins, human microRNAs, human messenger RNAs that are just circulating, in the hopes of finding a set of proteins or microRNAs that are specific to having cancer and hopefully sensitive enough to detect if cancer is present. Okay, those are two kind of different questions, but each are meaningful, because even if you have something that will tell whether it's breast cancer or not, that's valuable to a woman who's about to get a biopsy, right? The advantage of this is not only can I search for markers of breast cancer, but we can also, because we're growing these tumors, and then we can put those in if they grow, then we can put them in a series of mice and test therapies, right? So I've developed a number of collaborations, and these happen because when you're doing your research, you go out and you talk to people, and you give presentations like this. And your student talks to their student, or they see you talk and say, hey, I could use that for my research. Because networking isn't just something that's handy politically, it's also handy for your work. It's handy for finding scholarships. It's handy for finding out which professor you really don't want to take genetics from, right? So one of those projects is with Brent Reynolds and Loic Delaroe, and I'll go into that a little bit. So they had come up with, Brent's an interesting guy. He did his Ph.D. and discovered the way to identify the neural stem cell. It's the stem cell that can differentiate to form your brain, right? Pretty cool stuff. Founded a company, became the chief scientific officer, then became disillusioned with the whole pharma business, and went and did studies in Eastern medicine. And along the way, his wife developed breast cancer. So he became more interested in Eastern medicine, but he realized that his passion really was doing the neuroscience. So he studies brain tumors as well as some breast tumors. And he had his people work on combing the literature for herbal compounds that are used in Eastern medicine that actually show some efficacy, at least in a dish, at fighting cancer cells. Then he narrowed those down to the ones that you could actually get the concentration that you can get that kills the cancer cells. You could actually attain that in humans. That narrowed it down surprisingly. The degree to which that narrowed it down was from thousands to around 19. And then he started working with various combinations of those in tumor xenografts using cell lines. So we take, anybody heard of Henrietta Lacks? Anyone? Okay, my people have. So Henrietta Lacks was a very unfortunate woman who in the 30s, 40s, I can't remember the exact time frame, developed cervical cancer, okay? And back then, some of the restrictions on clinical science were not present, and her physician grabbed some of her cervical cancer and started growing it in a dish. And that was the foundation for what became the HeLa cell. So at least had the virtue of naming the cell line after her. But it was the foundation for much of cancer research used in research labs worldwide. She died shortly after he harvested cells. And those HeLa cells will grow in anything you put them in. Put them in a dish with media, put them in a mouse, Put them under your skin, they'd probably grow there too, so I wouldn't suggest it. But that discovery that you could do that was also the foundation for sort of an expansion of the use of cancer cell lines to get at the biology of cancer. Take it out of the human, grow it, and figure out the cancer biology pathways. Well, if you put them in mice, it's a little closer to a human environment than a dish. In fact, it's a lot closer. And what he found is if you use this combination, you could inhibit growth. And they each inhibit several pathways involved in cancer. And they're fairly well known. Curcumin is part of turmeric. It's the yellow stuff in all the Indian food, right? EGCG is part of green tea. And sulforaphane is anytime you eat a cruciferous vegetable. That's asparagus, broccoli, cauliflower. It's got a fairly high content of this. So he showed that it inhibited growth in his cancer cell lines. So we've tried it in a few of our primary patient-derived xenografts. So our xenografts are straight from a patient, straight to a mouse. There's no time growing in a dish. The dish isn't exactly like a mouse or like a mammal at all. So grow them in a dish long enough and the tumors change. Grow them in a mouse, it probably changes too, but it's a little closer to a human than it is to a dish. And sure enough, with this combination, it didn't do a lot added to chemotherapy, but it wasn't any worse. And in a few lines, there was a suggestion that maybe it was better. And about the time we were doing this, I heard from Brent that in his glioblastoma lines that the true neural stem cells actually survive better. You kill the cancer cells, the neural stem cells survive better. And I had a few patients who were on this chemotherapy agent, and when they're on this chemotherapy, there are toxicities that go along with chemotherapy. I mean, oncologists, we're balancing poison to benefit, right? We're killing cancer cells, hopefully not killing you. This particular chemotherapy causes numbness and tingling, loss of innervation, and pain through a process we call neuropathy, which is neurons or nerves having bad stuff, pathy, or pathology. And so I said, well, let's test the mice and see if they're developing neuropathy. And it turns out, and the way we measure neuropathy usually is the way we do for diabetics, which is we have this little filament, we press it against the bottom of the foot until it bends. That means it's a certain pressure that we're exerting. Say, can you feel that? And they say yes or no. Or which foot am I doing this on, left or right? Well, if we do that to the mice, we watch for the mice to go, I don't like that, right? And in the control mice, the number of times that they lift their foot is about half the time. If you're treating with the chemotherapy, it goes down to about 20% of the time. And on the combination, you start to get up closer back to normal. And that's with both the initial chemotherapy, but also a different chemotherapy that also causes neuropathy. People on these drugs also are hypersensitive to cold. So they reach in to grab something out of the refrigerator and it sends excruciating tingles and sharp pains, like if I hit your funny bone. And it seemed to protect from the cold extremely well. Additionally, it seemed to protect a little bit from anemia, which contributes to fatigue that most people get on chemo. So we're evaluating a lot of things that are related to that and looking at different targets of compounds that affect different pathways of cancer biology. For that combination, we're also trying to move to clinical trials to test it. So this is a bench to bedside to bench to bedside kind of story, hopefully. So I couldn't do any of this if I didn't have money. So UF, through the Gatorade Trust, provided the startup funds to open my lab, and still in part pays Kelsey's salary, as well as the Florida Breast Cancer Foundation, the Gatorade Trust, the Department of Defense, and the Cancer Center. And then I have collaborators, the Reynolds Lab, the Frost Lab, the Law Lab, Brown and Mohammadzadah labs. And then these are various prior technicians in my lab, grad students, and then my Army, which is here. And I brought a couple of members of my Army, Kelsey and Elizabeth, because this is about you all and showing that there is a path forward. You just have to set your goals, and you need to find ways to get there. And they came and started working in my lab as undergrads. Kelsey is now graduated and is working as my lab manager in transition until she goes to med school. So I'll have them stand up and you all can ask us questions.